Yes, so thank you for the introduction, and also thank you for inviting me to give this talk today. And as you've already heard, I will be talking about the subsoil. So for those of you who are not that familiar with soils, I've brought you a picture. Sorry. Which screen do you see, actually? Sorry. Now it works. So if we look at such a soil profile, we would call the upper part that is in the darker color here, that is the topsoil, and in agricultural soils that usually corresponds to the tillage depth or the plowing depth. So the lower boundary for this is somewhere around 30 centimeters typically. And anything that is below that is what we would call the subsoil. And we've also already heard in the previous talk something about the subsoil heterogeneity. But what I want to focus on today is more what are the resources that can be found in the subsoil. Our colleagues from the Thunen Institute in Braunschweig, they've done an inventory of agricultural soils in Germany, and they have analyzed different resources such as water or different nutrients. And generally from that inventory, what we can say is that around half of all the resources that we find in the soil are in the subsoil, so in this lower part of the soil profile. But also what we know from this inventory is that around half of the subsoil resources are actually in or below root restricting layers. Root restricting layers in this context means that, for example, the soil is so densely packed that the roots cannot grow through, and they cannot access the resources that would, in theory, be available there. So what I want to address during my talk today are basically three different questions. The first one is, how are these subsoil resources affected by what we are currently already doing in terms of agricultural management? What can we do to alleviate these root restrictions to root growth? How can we make the subsoil resources more accessible? And eventually, we will also need some kind of tool to actually see what is going on for the plants. So the roots are usually considered the hidden half of the plant, and we somehow need to find out what is going on down there, especially in the subsoil, to see how successful we are with our management. And for the purpose of my talk, I will be mostly focusing on the phosphorus, so on this part of the graph. Right, so in order to see what current management is doing to subsoil resources, or in terms of including subsoil resources, we can go to long-term field experiments. Within the Soil 3 project, we have sampled quite a number of long-term field experiments. So these are experiments that have had the same management for a number of decades. I don't want to show you all of those experiments that we sampled. I'll just show you 3 examples. For 2 examples, we will look at phosphorus fertilization, and then also 1 example on nitrogen fertilization. Right, so starting with phosphorus fertilization. For this, we will look at 2 long-term field experiments, one in Tyrol, which is close to Berlin, and this field experiment was started in 1937, and the other field experiment is in Gießen in central Germany and was started in 1954. Both experiments were sampled in 2016, so we have something between 60 and 80 years of constant management leading up to our sampling. When we want to know what the effects of p-fertilization, we can look at the phosphorus that is extracted by calcium acetate, calcium lactate extraction. That is actually the method that is also recommended by German authorities if you want to determine how much phosphorus is available in your soil and how much fertilizer you still have to add. So if we look at this fraction of plant available phosphorus, and we compare the treatments in the field experiments that have received phosphorus containing fertilizer over those almost 80 years in the case of Tyrol, and compare those to the treatments that did not receive any P fertilizer, we see that, of course, those treatments with fertilizer have significantly more plant available P than those without. And this is true not only for the topsoil, but also for the subsoil part of the soil profile. If we now look at the same treatments in the field experiment in Gießen, we see there as well an increase in plant available phosphorus in the topsoil, but in the subsoil, this difference is, well, marginally visible, I would say. This is not so much related to the different age of the field experiment, but it's probably more a texture effect. So in Gießen, we have a very clay-rich soil, very fine-textured soil that has a high capacity for P fixation. So most of the fertilizer that is applied to the soil will be directly absorbed onto the soil particles and will no longer be available for the plants. Whereas in the sandy soil that we have in Tewel, most of the fertilizer will remain in plant available form and will probably accumulate in the soil. So first of all, P fertilization increases P availability. But what I would actually be more interested in is whether this is just a passive accumulation of fertilizer over several years, or whether this fertilizer is then also included into the biological turnover of phosphorus at these sites. So whether it is used by microorganisms or also by the plants and recycled through the soil. In order to show you how we can do that or how we can test that, I have to take a short excursion into the theory of isotopes, but I promise I will keep it short. So if we look at phosphate, this is what you see here. This is a phosphate ion, so this is what the plant would typically take up from the soil. We would, of course, be interested in the phosphorus itself, in the central atom. But unfortunately, phosphorus occurs in nature only in the form of phosphorus 31 isotopes. We can artificially produce phosphorus 32 and phosphorus 33 isotopes, but these are radioactive, so they are not that well suited for application in a field experiment. But what you can also see in this phosphate ion is that phosphorus usually occurs in combination with oxygen, and oxygen does have 3 stable isotopes. So we can now use the ratio of the 2 most abundant isotopes, the ratio of oxygen 18 to oxygen 16, and we can use that as a tracer of biological P turnover in the soil. And that is conventionally designated as the delta 18O value. Now, how does that work? From a number of referencing studies, we know that for the parent material, for phosphorus contained in the parent material, we would expect rather low isotope values, somewhere in the area of 10 per mil. By comparison, phosphorus from organic matter or also phosphorus from mineral fertilizers has rather high isotopic values, somewhere around 20 per mil. So these are the end members that we put into our soils. And the only way that the oxygen isotopic signature of these phosphorus sources can change is by microbial activity. So what is happening there is that when microorganisms take up phosphorus and include that in their metabolism, for example, in the energy metabolism, so transformation of ATP to ADP and back to produce energy, there is an enzymatic exchange of oxygen isotopes between the phosphate and the water. And if this happens over and over and over again, eventually we will have an equilibration between the isotope ratios in the water and the phosphate. And the exact value of that equilibration is temperature dependent. So if we know the temperature at which this process happens, and we know the isotopic value of the water, we can make some predictions for the value that we would expect to see in the soil if there is intensive biological recycling. So if we now extract our phosphorus from the soil samples and plot the isotopic values that we see in the phosphate, we would interpret anything like here in the topsoil, anything that falls within the range of the equilibrium to indicate high biological turnover, whereas values that are close, for example, here in the subsoil, close to the value of phosphorus in the parent material, that means there is no or only very little biological turnover. So basically what this is telling you is whether the P is, let's say, biologically active, whether it is taken up by microorganisms and recycled, and anything that is biologically active in this way should, in theory, also be available for uptake by the plants. Now, if we go back with this information to our field experiments, what you see here are, first of all, the isotope values of the treatments that do receive P fertilizer. And if I now put in the reference values that I've shown you before, so the red one is the range of equilibrium values that we would expect from intensive turnover. We also have the reference values for the organic phosphorus in organic matter or in fertilizer, and we have the reference values of the parent material. And from this, you can see that for the treatments that do receive P fertilizer, we have a rather high biological turnover of phosphorus throughout the complete soil profile. So that does include the subsoil. By comparison, if we look at the treatments that did not receive P fertilizer for 60 to 80 years, these have, here in Tuval, for example, significantly lower isotope values. So this indicates lower biological cycling. So if you were expecting that the subsoil will be able to somehow compensate for missing fertilizer or missing nutrients in the topsoil, you might be disappointed. Rather, on the opposite, we would suggest that only if you do add fertilizer, you can stimulate biological cycling throughout the whole soil profile. So you stimulate including the subsoil into nutrient acquisition strategies. We see a similar effect also in Gießen, but with a lower magnitude or with a lower difference between treatment, smaller difference. And again, this is probably an effect of the differences in soil texture between the two sites. But generally, again, P fertilization enhances P cycling in the subsoil. Now you may say, well, it's not that surprising that P availability and P recycling increases if I add phosphorus fertilizer. That's true. So to be a bit more convincing, I will show you a different example on nitrogen fertilization. For nitrogen fertilization, we move on to the field experiment in Wolfratshausen, which is also in central Germany. It was established in the 80s, so we have about 30 years of constant management up to our sampling. And the soil is a loamy, lush soil, quite similar to the soil that we find here in the Bonn area. In this field experiment in Wolfratshausen, we have different levels of mineral N fertilization. There are treatments that do not receive any mineral N fertilizer at all for the past 30 years. And there are treatments that receive 100 or 200 kilogram per hectare and per year. And again, first of all, we look at the stocks of plant available phosphorus, and you see the highest stocks of plant available phosphorus are in the treatments that did not receive any N fertilizer at all. So apparently, with the missing N fertilization and very weak plant development, very weak plant growth, most of the phosphorus that is in the soil just accumulates and is not used or not efficiently extracted by the plants. With increasing N fertilization, the phosphorus stocks in the soil decrease, both again in the topsoil and in the subsoil, compared to the non-fertilized treatments. So with increasing N fertilization, we should also stimulate a more efficient recycling of phosphorus resources. If that is the case, then we should also see this in the isotopes, in the isotope values. And indeed, when we compare treatments that did not receive any N fertilizer to those that did receive N fertilizer, we see higher isotopic values, and also, especially in subsoil, higher isotopic values in the fertilized treatments than in the non-fertilized treatments. So similar to what I told you about phosphorus fertilization, also nitrogen fertilization stimulates that plant growth and microbial recycling of phosphorus proceeds to greater depth within the soil profile, so more of the subsoil is included. And I can show you also similar examples or similar results for experiments where irrigation was applied or where lime had been applied, so pH management. Basically, the outcome always is that whatever you do in terms of topsoil management or whatever kind of management you apply at the top of the soil surface will also have an effect on more downstairs parts of the soil profile. So if we go back to the questions that I set out in the beginning, the answer to the first one is a clear yes. Whatever you do or whatever we are already doing in terms of management strategies already does affect how much of the subsoil resources can be potentially used and how much of that is biologically active, as you would say. But of course, we still have this problem of the root restricting layers that mask about half of our resources that could potentially be accessible. If we want to somehow remove these root restricting layers, we basically have two options. One option that was very popular, especially around the middle of the last century, is deep plowing. So in the 1940s, 50s, 60s, this was done rather frequently in Germany. You see here in this picture, this gigantic moldboard plow, which would loosen the soil and turn it down to a depth of 1 meter, sometimes even. So we go far beyond the current plowing depth of around 30 centimeters. And when we look at these soils today, we can actually still see that. So the dark strips that you see here in the soil profile, this is actually the former topsoil that was then completely turned around and is now in the subsoil of these soil profiles. Within the Soil 3 project, we also analyzed some of these sites again. So they had received deep plowing in the 1950s or 1960s, and we wanted to see what are the long-term effects of these, well, let's say rather brute force strategies of deep plowing the soil. So these are the 3 sites that we looked at. All 3 are in northwestern Germany and Lower Saxony. And if we look at, again, the stocks of plant available phosphorus, and we compare the part of the field that had been deep plowed in relation to a neighboring part of the field that had not been deep plowed back then, we see an increase in overall phosphorus stocks. And this is also, or especially apparent in the subsoil, where now the former nutrient-rich subsoil is included, so which has been the former nutrient-rich topsoil has been included by turning around the complete soil profile. So apparently these nutrients are preserved there, and this also still today, even 60 years after this has been done, we still see effects on the grain yields at these sites. This is for the cereals that were grown there. However, you see both for the nutrients and for the grain yield that this effect is really site-specific. We have the site in Elsa, which shows almost no increase or no difference in yield, and also no difference in nutrient stocks compared to the site in Essemühle, where the difference is really huge. So as a kind of conclusion from this mechanical loosening of the soil, we can say, yes, deep plowing can have long-term positive effects. You can loosen the soil and provide the plants with more nutrients, but the effect is really site-specific. And we also know of some sites where this positive effect was lost or was gone after a year or two, and the situation afterwards was really worse than it was before. So this mechanical strategy of removing root restricting layers should be considered, or should be really carefully considered. If you do not want to use this really strong method of turning the subsoil, which also requires a huge input in terms of machinery, in terms of fuel costs, whatever, we can also use biological helpers, I would say. So for example, if you grow tap-rooted crops, such as alfalfa, in your field for a year or two, the tap roots will create these large-scale pores. And these pores will then be used by the roots of the subsequent crops. So you see here, these are, again, some cereal roots that are growing through the pores that are left behind by the alfalfa. If the bulk soil is too densely packed for the roots to grow through, these pores provide a gateway for the roots to circumvent this compaction and access resources that might be located further down below. And since these pores seem to provide some preferential root growth paths, in a former project, we analyzed the properties also of these pores. And we found that the walls of these pores are really enriched in nutrients compared to the bulk soil, or the normal soil that you see there in the middle. These higher contents of plant available nutrients are probably derived from the root residues, so the decomposed alfalfa root that was previously in the crop, or also from biological activity, so earthworms moving through the pore and incorporating nutrient-rich material. Sorry, that was supposed to be the other way around. If we also look at the isotopes in these pores compared to the bulk soil, we see again a higher indications of higher biological activity. So based on that, you may suggest that these biopores present hotspots of nutrient acquisition. There is a lot of nutrients and they are easily available to the plants. But what we also see, and this is now what we are continuing in the context of PhenoRob, is that if you grow plants in soil with pores compared to plants in soil without pores, that the plants that have pores available invest much more into root biomass. So they have a much higher root to shoot ratio than the plants growing in soil without pores. And we also later, after the experiment, we inspected the roots that were growing in these pores by microscopy. And we saw that for some roots, we clearly have soil contact. So you see some soil particles here in the microscopy image. And also all these little green dots that you see in the picture are bacteria that are on the root. They have incorporated the fluorescent dyes so that we can visualize them. So this root has clearly been in contact with the soil or with the pore wall, and then can also take up the nutrients that are available there. But we also found roots that are nearly clean, that are really blank, do not show any soil or any soil particles or any bacteria. So these roots were growing in the pores, but they did not have any contact with the pore wall. And in this case, we would then say this presents more of an empty investment into root biomass, because the plant produces a lot of root, but it does not get any benefits of getting the nutrients from the pore wall. So with respect to that second question, yes, we can do something about the root restricting layers, but it is always a cost-benefit trade-off, both for the farmer, who has to consider how much he or she wants to put in terms of machinery, fuel costs, and also in including that into the crop rotation. But it's also a cost-benefit trade-off for the plants. How much additional root biomass do I have to produce to access those extra percent of nutrients? So from those first two chapters, first two questions, what we see is, yes, there are ways to include or to target the subsoil in our management strategies, but the effects can be really variable, or the results can be really variable. So we would somehow need a tool to control for how effective and how successful we really are. For this, I would like to go back to the oxygen isotopes once more. So I showed you, or explained before, that in the soil we have our end members nicely separated, and we can calculate the value that we would expect from microbial cycling so any kind of biological interaction. But this process that is driving the microbial cycle or the exchange in oxygen isotope is unfortunately not exclusive to microbial cells. The same thing is also happening, of course, within plant cells. So also plant cells in their energy metabolism do this turnover and exchange of phosphorus. Now, in the plants, that actually makes me a lot less happy, because that might also mean that whatever the plant has taken up in phosphorus, I cannot see it afterwards anymore, because this original isotopic signal of the source is overprinted by what the plant is doing internally in itself. So to test if that is actually a problem or not, we conducted another pot experiment with springweed, very young springweed, as you can see in this picture. That was at about 10 days after germination. And these springweed plants were then supplied with a nutrient solution that contained unlabeled phosphate. So this is phosphate that is isotopically similar to the phosphate that we also see in the soil. And that is the phosphate that I want to trace then into my plant to see if or where or when any exchange with the water occurs within the plant. We added labeled water to the nutrient solution. And then we monitored the oxygen isotope values in the plant shoot and in the plant root over the next 10 days after the addition. So the green dots here indicate the the shoot values, and the orange dots indicate the root values. And you see that both for roots and shoots, we have an increase in isotope values, so there is some exchange happening already within the first 3 to 5 days. If I now add the corresponding water values, we can also again calculate the value that we would expect if the turnover has been complete. And you see that especially the values that we get from the shoot material, so these green dots here, they are mostly within the range of that equilibrium. So whatever phosphorus has been in the shoot or has been transported into the shoot is completely overprinted by the internal cycling of the plant. In the roots, however, we do find values that are still about 30% below that expected equilibrium value. So that means, in reverse, some of the phosphorus, some fraction of the phosphorus in the roots still retains the original signature of the phosphorus source, so the fertilizer in this case, that the plant has taken up. To get a somewhat better idea of what is happening there and where these 30% are coming from, we then built a model together with Andreas Schneff, and this model was supposed to replicate the experiment that we were doing. I do not want to go too much into the details of this model, but what I would like to highlight is that we did allow for oxygen isotope exchange both in the root and in the shoot, and we also had to allow for back transport of phosphate that was recycled in the shoot via phloem transport back into the root. And eventually, with that model, we were able to replicate the experimental results of that greenhouse experiment quite nicely. So the lines here are the outputs of the model, predicted trends in isotope values from the model. And then by turning on and off different components within the model, we were able to understand better which processes actually contribute to these values that we see here in the roots. And we've eventually found out that actually this back transport of recycled phosphorus from the shoot was more important than the direct exchange of oxygen isotopes within the root. So like I said, I do not want to go too much into the details of that. The general message here is just that from the combination of isotope data and the model, we were able to figure out where the phosphorus that we observed in the root was actually coming from. So we do have some information on the phosphorus source. Now, what do we make of all of this? How does it help us to evaluate how efficient our subsoil management is? If we imagine a plant growing in the field, we can also imagine that any phosphorus that is taken up anywhere within the root system has to bypass the hypophytals, so the upper part of the root system, to be transported into above ground plant organs. Now, what we're also doing in PhenoRob and in CP3, what I didn't show because of the time constraints that we have, is we're testing the use of endoscopy for a targeted inspection and a targeted sampling of the root system. So if by that targeted sampling we would analyze the upper part of the root system that somehow contains all of the information about phosphorus uptake in lower parts of the root system as well, we can analyze those samples again for the isotopic signatures, similar to what we did in the pot experiment in the greenhouse. These data we can then bring together with the isotopic values that we know from the soil profile. So eventually, if we back that up with some isotopic mixing model again, we should be able to determine which part of the soil profile contributes to what extent to the overall phosphorus nutrition of the plant. This whole concept here is actually something that is already done, especially if you analyze water uptake of crops, but for phosphorus, nobody has dared to do it so far. So I do think we still have quite a long way to go until we can eventually use that in field experiment, especially this part here of what is happening in the plants, under which conditions. That is something that still needs to be constrained a lot better. But I would say the tools that we need to do this at some point in the future are already available. So eventually we should be able to determine how successful we are in including subsoil resources into our management without having to actually dig up too much of the soil profile. So with this, I would like to thank you for your attention, and I'm happy to take questions. Thank you.